Hi, I'm Erin McDermott and I work as the genetics technician in the NC State Apiculture Lab. I primarily work on a collaborative project studying the effect of pest management practices on honeybee health, specifically how it affects viral loads. Part of our effort with several other universities for this project is studying which management practices will help keep healthier bees and help them more successfully over winter. We analyze these samples for viruses via qPCR and add them to our overall dataset. I also assist with our Queen and Disease Clinic. This clinic is a service offered to beekeepers who want empirical measures for bee health. We typically work with queens. Queens are particularly important because their health and viability affects the entire colony because they're the only reproductive female. We can test for reproductive viability of queens and drones via a number of morphometric measures as well as sperm viability, and we can also test for viruses and other pathogens via qPCR. We're also able to use microsatellites to determine genetic variability in a colony. We can measure the number of drone fathers, letting us know what kind of genetic diversity we have in a colony. Higher genetic diversity can help bees respond to stressors like pathogens and viruses. Applying these next-gen techniques to real-world problems help us answer a lot of questions that beekeepers and scientists have had for a very long time. qPCR allows us to absolutely measure viruses and pathogens, which tells us not only whether or not a colony is sick, but how sick it might be. Knowing how infected bees are helps us discover the levels at which these pathogens start to have real impact on health and ability to overwinter. That's something that we're exploring right now with our dataset of over 900 colonies. We have a lot of statistical power with a dataset of this size, and we're hoping to get down to the answer to the question, how sick are my bees and what effect will that have on my colony? Our process starts with a tube of frozen bees, collected either on dry ice or in liquid nitrogen, and kept at -80 to preserve RNA. We extract under a fume hood, turning whole bees into a usable sample. We can then synthesize cDNA, load it onto a qPCR plate, and measure absolute load of viruses. We typically test for acute bee paralysis virus, black queen cell virus, chronic bee paralysis virus, 2 different strains of deformed wing virus, Israel acute paralysis virus, Lake Sinai virus, and Nosema and trypanosomes, which are other pathogens. There is a growing body of evidence that these diseases can have significant impacts on honeybee health, and we're already discovering some exciting trends in our data. We identified black queen cell virus and Nosema in more samples than any other pathogen, and out of our 915 samples, only 43 were completely clean, supporting the idea that Varroa is facilitating the spread of viruses and other pathogens in our bees nationwide. We're hoping that as we continue to analyze these samples, we'll learn more about what these diseases are doing to our bees. As we continue to develop our Queen and Disease Clinic, we're continuing to add new products to help beekeepers. We have some really exciting projects in the works, dealing with measures of viability and even a type of pedigree that can tell us more about a colony's genetic history. We hope that beekeepers at all scales will be able to use the information we're providing to inform their management practices and not only raise healthier bees, but also help control the spread of Varroa and the diseases that they carry. If you want to know more about the services that we offer, you can check our website. And if you'd like to keep up with what we're doing on the day-to-day, find us on Facebook.